Dear students, we are going to discuss PyMol, how to install it and how to use it for three-dimensional structure visualization. So what you have to do, you can search for the PyMol in the Google and you will come up with these search results. You can go for the registration for educational use. PyMol is free for the educational use. So you can go and register yourself. Here you need to fill out this information and it will take you to the next page, which is this one. And you fill out this and press I agree. And at the end of this, it will send you an email, which would look like something like this. Can open this email, and at the end of this email, you will have this link and username and the password. So if you click on this link, it will take you to this page. It would first ask you for the username and the password. I already had PyMol installed on my computer, so it didn't ask me for all that information. So you can select the version suited to your system. So my laptop is Windows 64-bit, so I downloaded this version. So it would be downloaded as an exe file, and you can install it by clicking on this, something like this, and run. Okay, I already have this on my computer, so I don't need to do that, but you can do this. Okay, what next we need to do, once you have installed it, you can open the PyMol. Here I can search it, and so it would open something like this. Okay, so now we have the PyMol installed on our computer. What we want now, we want a macromolecule structure, say for example a protein structure. So from where we can get a protein structure? We can get the protein structure from protein data bank. Data bank is a database which stores all the published macromolecular structures. So you can go there and you can select the structure, search for the structure of any protein whose structure has been solved by NMR, X-ray crystallography, or mass or electron microscopically. So here I'm going to search for a protein which is called as RAL-A. This is a RAS family protein which is involved in cancer. And here are some results. Each protein in PDB or any macromolecule in protein data bank has a code which is specific to that protein. Say for example, I can download this protein, which is a NMR structure of RAL-A. So this page would give you some of the more information about this structure, the publication. It can give you the information how many chains are there. So there is only one chain, the sequence, how many amino acids are in this sequence, the organism. So it's a human protein, and mutations, if any. So this protein has one mutation, and then some more data. So it can gives you the link to the other databases, which is a UniProt. So this is the ligand bound to it, which is GNP, and the other ligand bound to is this protein is magnesium ion. Okay, so what we want to download the structure of this protein and we want to visualize it in the PyMol. All right, so you can click download files and you can download this file in the form of PDB format. Okay, so this would be downloaded. Now you can go to the PyMol. So this was a browser we opened earlier. We can go here. And we can go to the downloads where our protein was downloaded. So we can open this one here. So it would open something like this. So this is our protein, and we have it in the PyMol now, right? So let's see, if I want to visualize the sequence of this protein, I can go to display and I can visualize the sequence of this protein. Okay, so it's at the top, the sequence is being displayed. How you use this browser? With the, if you're using a mouse, the left, from the left finger, you can move it something like this. From the center button, you can move this molecule at different position in this window. And from the right button, you can zoom it on, zoom it in and zoom it out. And if at any point you lose your protein, you can press a reset button here and it will reset your protein to the original position. Okay. We also have some control panel here and it actually displays our proteins. So what we can do with our protein here, we can say, for example, I don't want it in this stick kind of a diagram, which is not giving me much information. So I can hide. So here we have some action button. A stands for action. There are various actions we can do. We can show our protein in the form of cartoon, ribbon, and sphere, surface view. So disulfide bonds, we can show various formats and parameters of our protein from this button. Hide button shows where we can hide everything which was shown in the in the S or the S panel. And L would be something we can label. We can label the residue, chain, atom, element, segments. And from the C button, we can color them differently. We can use various colors on our selections, on our protein. So first of all, what we will do, we would hide everything and then we would display our protein in the form of cartoon, which is more of a common shape. We visualize our proteins in our books and presentations and all that. So you can see this protein has alpha helices, it has a beta sheet, it's a globular protein. Okay, and we can show here we got the cartoon diagram of our protein. We can color it differently if we want. Say, for example, I want it in blue, I can color it blue. See, I want it something like yellow, so I can color it yellow as well. You can use even the different shades of the given color. So here I have used a different shade of yellow. I can use green and various shades of green as well, and red, blue, purple, whatever color you want. You can color your protein according to your requirements, or whatever color you like. Okay, you can change the color from this panel. Let's go back. Now we know this protein has two ligands bound to it, but we cannot see them here in the cartoon diagram. So let's display those those ligands. So here we have GNP. We can show, so this is our selection. So any residue you select on this protein or any ligand you select, it will display as a selection here. Okay, so this is our selection. So our selection here right now is this GNP molecules, but it's not being displayed, only its position is shown here. So now we want to display this molecule in the form of sticks, for example, and now they are shown in green color, but I want to a distinct color, so I would color it something like red. Okay, so here my ligand is present bound to my RAS family protein RAL-A in the stick diagram. I also want to display the magnesium ion, so I can select the magnesium. Now this selection represents the magnesium, this selection. Okay, and this is a whole protein. Okay, if you display, you click on it, this button, you can hide everything almost from this window, and you can just click it on again and it will come back. So now I want to this magnesium ion as a selection here, and I want to show it in the sphere, and I want to show it as a yellow sphere, for example. Okay. So now this is my protein and it has this cartoon representation, and it is actually been shown as a ligand. The ligand is shown as a stick diagram, and another which is magnesium ion, which is it's shown as a sphere. Okay, so I know the ligand position, I can move it around, I can visualize it. You can store this if you want to store the it this if you have done all this, you want to store it as it is this PyMol session, you can go and you can save the PyMol session. Okay, say for example, I can save it as RLP. Okay, yes, right. So the my session is stored. Also, I can take the images. So, for example, in PNG format, if I want the image, I can store the image in the PNG legend found format. Okay, so this will be saved as an image. Okay, uh, it would always be showing here for the educational use only in all the images and all the sessions, because our PyMol, if we want to use it for any commercial purposes, for any industrial use, or for any publication purposes, you need to purchase the license. And for the educational use, it is free. So it would always be displayed here as for the next educational use. Okay, what else you can do? You can display, you can change the background. So if you can make it white, you can make it light gray, you can make it dark gray, whatever color you like, you can make it that color, and depending upon your requirements. And so you can do that here. Okay. There are a few more functions, but right now the functions we are more interested in. Say, for example, we want to visualize what are the surface charges of my protein. So we know that a globular protein has a hydrophilic kind of a surface, so it must have some charges present on its surface. And I want to visualize what kind of charged surface my protein has. So what I can do, I can go to this action button and I can go to generate, and this would can generate an electrostatic map in the vacuum. So although the electrostatic map in the vacuum wouldn't be exactly the same, what are the surface charges is present in a in a solvent or in water, but it would still give us some information about the protein, about the surface charges or the surface properties of this protein. So we can do that here by going to generate vacuum electrostatic and protein contact potentials. All right. So it would take some time, but let's wait for it. So now we have this protein molecule, and this is a surface view of our molecule. all right? And the red region is showing the negatively charged part of the protein, and the blue regions are showing the positively charged regions of our protein, and the white ones are the neutral surfaces. So you can visualize. So this is our protein molecule, and this is basically what I believe this is our pocket where the our nucleotide, our ligand binds. So you can see here. So this is our ligand binding pocket, and here we have other surfaces. So you can see the surface charges of a protein molecule using PyMol. Okay, this is done. So now we can actually go back, and we want our protein back from this. So this was the surface views. We don't want this right now. So this is the scale shown here. Here is the three-dimensional coordinates of our protein molecule. So this is our here. When you show the three-dimensional coordinates, you can visualize here the pocket, ligand binding pocket, and you can see how our molecule, which is a GNP, bound to our protein, which is it has negatively charged phosphate groups, and it has a magnesium ion. So you can see which is positively charged. So how it is interacting with the protein surface. So here all the protein surface which is interacting with the GNP is positively charged, and the side of the pocket which is interacting with the magnesium is negatively charged here inside this pocket. Okay, and now we can hide this by clicking on these buttons on the control panel, and your protein will be back. If your protein is not back, you can press on this one and it will bring back your protein molecule. What else I want to do? Say, for example, I want to look into the interaction of this amino acids which are interacting with this ligand. So we can go to this action button, and what we can do is we can present the ligand in so here is your ligand, and it is representing your ligand interactions of your ligand with the amino acids of your protein. So you can visualize your ligand somewhere here. So this is basically GNP, and this small green dot here is representing the magnesium. So it is basically showing the interactions of the ligand with the magnesium ion, and rest of the protein is being shown as a stick diagram here. So now you can also record this picture, for example, in the form of an image. Okay, now I want to hide this as well. And what I want is I want to present my ligand, but ligand side as well, in the form of a cartoon diagram. So now what I have here, so I have rest of the protein in the form of a cartoon diagram, but the amino acid who are interacting with my ligand are shown as a stick diagram. Okay, you can see this here. So all the region which is involved in the interaction with the ligand is shown as a stick diagram, while the rest of the protein is being shown as cartoon, and you can see the interactions of the ligand with the amino acids. So these small yellow dots represent the interactions the ligand with the amino acids. Okay, so you can, and here it is showing the interactions of magnesium ion and the amino acids with these yellow dots, dotted lines. Okay, So by doing this, you can actually visualize the interactions, the protein molecule and the ligand in a detailed manner. Okay. So I can also store this as an image if you want. So you can store the various images you required using file and ng format action. Okay. All right. Okay. So now let's just hide this one and let's hide everything and let's show our protein again as a cartoon format. Say, here, I have my protein in this cartoon format and it's colored as a rainbow. And what I want now, I want to select certain residues and want to display them on this molecule. Say, for example, I want to display this region. Okay, so I can select this region and this is my selection. So it's come up here. You can name this selection. Say, for example, region A. For example, I want to visualize this region in detail. I want to color it, say, for example, red here. I want to show it as a stick diagram. So they would be shown as a, sorry, not as protein hide sticks, only the selected part in the stick diagram. So you can show, if you see, click on the selected part, you can show that part as a stick diagram. Okay. So yes, this is how you can represent your proteins and you can visualize various kind of structures you want, various regions you want to highlight, ligand binding. You can use all various kind of parameters or options available in the PyMol to visualize your protein and to visualize significantly important regions of your protein, the interactions of protein using this software. Okay. And what I want now, I want you to watch this video and follow all these steps and submit a report which you will prepare by installing PyMol on your computer and by visualizing any protein you want to select from the PDB. You can download that protein and just prepare various ligand interactions of this protein, the surface charges of that protein, and various uh, interactions on selected regions of the protein if they're interested. Submit a report me with the images on that after watching this video. Okay. I have another interesting protein with me which I can show you. That protein is actually, so let's go to another session. And what that protein is basically spike protein, which is spike protein of COVID-19. And I have this one here and we can display the sequence here. And this is basically a trimer. So that means 3 chains are actually interacting with each other. So we can go here, similarly hide, and we can show it in the form of a cartoon here. And what I want here, I want to select each chain and I want to color it differently. As I already mentioned, there are 3 chains. So let's select these chains. There was a command available to do that. This one. On another thing which about PyMol is you can use this command line. This command line A, and this is my selection. So chain A is selected. Now and you can see here till this point, and now the chain B is starting here, and these are the ligands bound to this molecule, this chain. So this is my chain A selected. I can color it, say for example blue. So all my chain would be A would be shown as blue. So let's chain B now. It is now, and I can say for example color it as magenta. Now my chain B is selected and colored. Let's select chain C. Chain A. So I have three chains, so chain A, B, and C here. And now chain C is selected. Let's color it yellow. And this is how. So this is a spike protein. It has a primer, and it has been shown here. And all three chains are shown as different colors in the cartoon format, and you can see visualize the interactions of this protein. So you would see like some of the proteins are monomeric, like RAS protein we just visualized earlier, but some are like trimer, tetramer, hexamer. So you can visualize various chains by using this method. You can hide these chains. Say for example, I can hide chain B, I want to hide chain C as well, and I only want to work and visualize chain A. I can do that as well. This is various regions of the chain A spike protein. Okay, so I can bring them back as well. For that, I need to show them as cartoon. Okay, so then they will come back. All right. So you can play around with the PyMol, and you can visualize the and study the three-dimensional structure in this software. All right.